Cordy.
Hi, my name is Yuvraj, and today we're going to be talking about geovirology. Our question is, does geographic proximity correlate with genetic difference? I worked on this with my two professors, James Godey and Dr. Tippett. I also worked on this with my partners Elizabeth Guzman, Madison Meldrum, and Alexis Shields. So last semester, we found a phage called Brock monster. Uh, and this was very unique to us because it was a direct isolation. So we thought that it would have a significantly different genetic genome sequence than our other phage outcomes that come from enriched isolation. So we decided to compare it with one of our first phages we found, which was McShee, and we found that that it only had a base pair difference of 1, so they were actually quite similar. After that, we started beginning thinking that maybe it is correlated to the distance that we found them because they were only found a third of a mile away. So we decided to check similar clusters on phage DB blast searches. So we correlated bit score and distance, and bit score was to show how close those phages were. For this slide just shows how Brock Monster was our first direct isolation that our whole college program has found. And once again, our whole thing was hypothesis was that they're going to be extremely different because one was direct isolation and one was enriched. And this was obviously not supported because Brock Monster and McShee were only one base pair difference apart. This fame map basically shows we have Brock Monster on top, while we have McShee on bottom, and they're almost identical. We can see that. Once again, so once we found that our McShee and Brock Monster were that similar together, we decided to find other pairs of closely related phages that have been characterized in the C phages program and know how far their isolation sites are separated by physical distance. To do this, we compared the bit scores of the blast searches, and we compared the distance on the GPS. So for our methods, we analyzed 283 microbacterium phages from phages DB. We then ran phages DB blast and found the closest relative phages. We then did a reciprocal blast, or we looked at the blast of that other phage we found and saw if our original phage lined up with it. After that, we extracted the GPS coordinates from the phages DB and plugged them into a website called bolter.com to get the physical distance apart. Finally, we plotted in the bit scores from a blast searches versus distance. These are results on this fitted line plot. We found that comparing the bit score to distance has shows a low correlation, but is, as you can see with the p-value, is statistically significant. With these results showing basically how almost there's little to no correlation of bit score and distance. Our future plans are to, in the following months, keep comparing and finding, keep comparing bit scores to distance in the following months of different clusters. And that's it. Thank you.